You said earlier that H. pylori affects 10 to 15% of the people who have it negatively. Is H. pylori a good or bad bacteria, and how is this related to personal medicine?
I think that Raquel can answer this question.
For me? Okay, so I think there's already been a little bit of discussion on that, but okay, so I don't know if you can see, but so my opinion with all the information I have and been studying H. pylori for about 5 years is that it's a good bacteria. So I think people infected with H. pylori will develop some kind of gastritis, which is like inflammation of the stomach, but that goes without any symptoms. However, and Joseph has mentioned this, 15% will eventually develop ulcers, and 2% I think will develop cancer. So what you can see here is that like a map showing the prevalence of H. pylori. The relevant information is that H. pylori has, the prevalence has dropped very significantly over the past years. So even though 50% of the world's population carries H. pylori, in the past it was about 90%. So for the 75% of people who will not develop any significant disease, what it's believed currently is that they have some benefits, and those are that they are protected from, I think it's asthma, obesity, and in general, diseases that have an inflammatory component. So the important message about this is that the 50% of us, because I think probably most of us in this room don't carry the bacteria, we have lost protection, and we are exposed to other kinds of diseases which have been increasing very significantly. And I think the current protocols is that if a person goes to the doctor and is diagnosed, or as an H. pylori carrier, is going to undergo antibiotic therapy to eradicate it. So this goes back to how health is influenced by the environment. So H. pylori is just one of the bacteria that we know that by not being present leads to other diseases. And as I said, it's just one of the bacteria that may be in this group.
To follow up on...
Your mic. Your mic is not on.
Oh, it's not on. Okay. To follow up on that question, how can we do better than just using aggressive antibiotics to treat H. pylori or similar pathogens? Sure. Because using strong antibiotic is probably not preferential in many cases. So can you elaborate on that?
So, I mean, I assume that everybody in the audience is aware.
You can repeat the question.
The question is, what are the alternatives to the use of antibiotics to treat H. pylori or other infectious diseases? So everybody's aware that excessive use of antibiotics has led to many other problems like antibiotic resistance. So in the case of H. pylori, I think it's more whether you want to treat it or not. In other diseases, like for instance, C. diff, Clostridium difficile-associated disease, the prevalence is increasing very significantly, is associated with, again, the use of antibiotics and people staying in hospitals. So an alternative to this is, for instance, using fecal transplantation. So it's basically restoring the microbial environment in the intestine. For other infectious diseases, like, for instance, influenza, for which we don't have effective antivirals, another approach is to use host-targeted interventions. So really target the host so that it can, in the case of influenza, as I said, you can get rid of the virus more effectively, or you can recover from the inflammation that is associated with it. So basically, those are the two ways.
Are there any other questions?
So you talked about both modeling and experimentation. in the presentation. So I was just wondering how modeling can help experimentalists and clinicians, and how can clinicians and experimentalists help the modeling?
I think that may be a question for Stefan.
At least I can take the shot at this, answering this from the modeling side. And I think you making a point, this point already in their question. You're saying how can the experimentalist help the modeler, and how can the modeler help the experimentalist? And I think this working together can address already some of the frustration we've heard earlier this morning about the modelers or the analysts complaining not enough data, and the other being frustrated that the data cannot be used. So in our case, it's important that we work together. We are working together in building the model. We are working together in designing the experiment so that the data we can get is valuable for the model. And once we have a model, we make predictions. And these predictions, in a good case, are validated. In a bad case, they are not validated. However, from my point of the modeler, the last one is the interesting case, because that tells me that my model was wrong and I have to work on it. And from this standpoint of publication, the first one is a good case. So, but in either case, whether that model is validated or falsified, the new data is used to improve the model. And with a better model, we have better predictions, better experimentation, and we increase the knowledge discovery as a team. That's how I see it.
I just have another question. So what types of models do your tools support?
We have actually a suite of tools which we use in the lab. There are process tools which are simulating processes like chemical reactions, gene expressions, protein folding, complex building. Those we would use a tool like Copasi, which is a standard tool available for download from VBI, and everybody can use it. That allows stochastic and deterministic modeling. Often, if you start building models, you have knowledge about interactions like upregulation, downregulations of genes, proteins. In that case, we have a tool which is called PlantSimLab, which is actually a discrete modeling tool where you just specify information of upregulation, downregulation, and maybe just on and off. So it's a very low-level tool to start thinking in a mechanistic way. And on the other end, we have a multi-scale modeling tool or agent-based modeling tool, the INISI tool suite, which has an HPC component, which we heard already about, which can do 10 to the power 8 agents. And here you see actually a visualization of such analyzed data or created data. So we cover at least a wide spectrum of models with our tools. And several of those are freely available.
Any additional questions?
Have you run any clinical trials to test your therapeutics or nutritional compounds?
Yeah, I guess I'll address that question. We've run a clinical trial that was funded by a company in Crohn's disease patients. Our earlier research focused on a target called peroxisome proliferator activated receptor gamma. That receptor is the main target of type 2 diabetes medications, one of which is the current drug used, which I compare with abscisic acid, and I presented in the previous figure. What we found is that that receptor could be modulated by fatty acids. And if we did so, we would improve outcomes, health outcomes in the context of Crohn's disease. So we ran a clinical study, and the preliminary data for that study was quite promising. The downside to that is that PPAR gamma activating compounds have kind of a negative reputation because the family of compounds that are used for type 2 diabetes have side effects which can be quite devastating, including myocardial infarction. So we decided to move away from the PPAR gamma pathway, and we've been focusing on the other targets that we've discussed earlier. Obviously, the opportunities that we see in terms of clinical trial are not just recruiting patients and assessing immunological outcomes and how those immunological outcomes correlate to health and disease, but going beyond that and running in silico clinical studies that are built on a strong immunology data so that we are able to predict the outcome of drugs in silico before we begin to recruit the patients. And maybe Peter can talk a little bit more about that.
Is on right now. So to expand on that, we are trying to use the results from the clinical study to generate in silico population or virtual population of patients with different degrees of the disease and apply these drugs on them so that we can better understand how the drugs behave on the population and also have more predictive power in terms of inclusion criteria, what type of patients are more likely to benefit from the drugs, and so on. So that's the next step to advance the clinical trials faster toward actually reaching to the real population. So to test the safety as well.
Okay, is there any other question?
How is NIMML's research helping to create new cures to human diseases?
Yes, and I think that we can recognize our limitations. NIMML is a research laboratory within a university setting. And so we are very well set up for studies that address mechanism of action, that discover new biomarkers and therapeutics such as DLA and CL2 to build mathematical models to validate those models. We believe that the next step for developing therapeutics needs to be done in organizations that have been set up as such. And in fact, there's a spin-off company that stemmed from NIMML in 2008 that has licensed the intellectual property from Virginia Tech in order to be able to advance some of the compounds into the market. And so it is something beyond the walls of this institution, but it's something that is going on, and it's going on very actively.
Great. Well, thank you very much, Josep, and the rest of NIMML. Please, round of applause for a wonderful presentation and a really informative question and answer session. It's very appreciated.